everyone, welcome to the 1000 MCQ series of GPAT examination. This is our exclusive course designed for GPAT aspirants. So stop dreaming and start doing. Coming to the first question of the day, geriatric populations should be included in the following phase of clinical trials. So what are the different phases? Phase 1, phase 2, phase 3 or phase 4. They are asking for geriatric populations, right? So if we see each phase in detail, phase 1, why we are using? We are using for safety and dosage testing, right? Phase 2, why we are using phase 2? For effectiveness and further safety studies, in which we will be evaluating the ADR also. Coming to the phase 3, in phase 3 what we will be checking the efficacy and as well as monitoring the other ADRs. And coming to the phase 4, this is a long-term study, post-marketing surveillance study. So we will be testing the long-term effect as well as the efficiency of this drug. So here, as the number of phase increases, the number of participants also increases. For example, in phase 1, it is 20 to 100 healthy volunteers. But in some conditions such as diseases such as cancer, we will be using the cancer patient instead of healthy volunteers. Now, coming to the phase 2, it will contain hundreds of people. Coming to phase 3, it will contain 300 to 3000 volunteers. And coming to phase 4, it will include thousands of volunteers because we are testing this after the drug enters to the market. Right. So the question is, geriatric population will be tested in which clinical phase? It will be tested in the phase 3, right? Phase 3 of the clinical phases. Coming to the next question, rabies vaccine living. See, whenever a question comes like vaccine living, it means it is live attenuated vaccines, right? It is obtained from or prepared from sheep blood, horse plasma, fertile eggs or mice lymph. See, rabies vaccine or live attenuated rabies vaccine is prepared by using fertile eggs, right? This from the fertile egg, we will do cell culture, and by using the cell culture, we will make this particular vaccine. So what will be the answer? The answer will be fertile eggs. Coming to the next question, in nail polish, following polymer is used as a film forming agent. Right, which is the polymer? Nitrocellulose, polylactic acid, hydroxypropyl methyl cellulose or HPMC, and all of the above. Right, in nail polish, film forming agent. See, nitrocellulose or cellulose nitrates, these polymers have the ability to form exceptional films. Right, we have to let's see some of the main characters of these polymers. First one is, it is a primary film forming agent. Film forming agent means it will dry quickly and form a film over the nail surface. Second thing is, it is having a quick drying property. After application, the nail polish will dry up very quickly. Next one is protection, which means it will protect the nail surface from the other external environmental stimuli. Next one is adhesion. Whenever we are applying this formulation of a nail polish, it will adhere to the surface of the nail. That is adhesion. Coming to the solubility, nitrocellulose can be easily solubilized in the organic solvent that we are using for making this nail polish, right? So, which is a polymer that is used as a film former? It is nitrocellulose. Coming to the next question, drugs in suspension and semi-solid formulations always degrade by? Is it first order kinetics, zero order kinetics, or second order kinetics, or all of the above? See, in suspensions and semi-solid formulations, degradation takes place by zero order kinetics. Zero order kinetic means what? The rate of degradation, means amount or the speed of degradation, is independent of concentration. As you can see here, even the concentration is increasing, there is no change in the rate of degradation. Right. While in other conditions, for example, in first order, if you see, as a concentration increases, as a concentration of the drug or the compound increases, the rate of degradation is also increasing. That is first order. So in semi-solid as well as in suspension kind of formulations, usually it will degrade by zero order kinetics. Why? Because the degradation is not only depending upon the drug concentration, but it will depend upon other factors such as light, temperature, etc. right? So the rate of degradation, it's constant and is not dependent upon the concentration of the drug. So what will be the answer for this question? It will be zero order kinetics. Coming to the last question, drugs following one compartment open model. One compartment open model pharmacokinetics eliminate by. Is it bi-exponential, tri-exponential, non-exponential or mono-exponential? See, in one compartment open model, we are saying whenever we are injecting a drug, it will distribute throughout the body. It will distribute throughout the body, right? That is open model of one compartment model. So this is an assumption. In actual scenario, this is not what happens. Now, when this distribute all over the body, we say that there is a single exponential decay curve that it will follow, which means the concentration of the drug in the body will decrease exponentially over time. Why? Because of elimination, right? And this will follow a single exponential decay curve, right? So that's why what will the answer? The answer will be mono exponential. Whenever one compartment open model is coming, the pharmacokinetics eliminates by using mono exponentially, right? So this is our exclusive course for GPAT, which we have designed for the GPAT aspirants. So stop dreaming and start doing. Hope you enjoy the video.